Alrighty, so today we are going to start talking about the process of fermentation. This is what happens when we don't have oxygen to generate ATP, to break down foods into energy. The plan is to talk about fermentation today. I'm going to hand out the lab we are going to complete tomorrow, so you can read through it, do the pre-lab questions after we finish this up. And then after we do the lab, there is no lab report for this one. There are like data analysis questions, but there's no lab report. We are going to start photosynthesis. And then we have like a week of photosynthesis activities and a photosynthesis lab. That lab will have a lab report. Does anybody have any questions about that kind of stuff? All right, then let's get started with fermentation. My goal for you is to be able to list the inputs and outputs of fermentation, just like we did with cell respiration, as well as differentiate between two types of fermentation that we're going to talk about: lactic acid fermentation and alcohol fermentation. So, to start us off, we need ATP without oxygen. And by we, I mean sometimes it's cells in our body, but most of the time they are single-celled or very small multi-celled organisms that carry out this process. So fermentation, as well as anaerobic respiration, fermentation is a subset of anaerobic respiration, enable cells to produce ATP without oxygen. They don't need oxygen as that like final electron acceptor because they don't often use an electron transport chain. Anaerobic respiration does technically, that's why how we get the respiration bit, but they use a different electron carrier. So they use something other than oxygen to create that last little step in respiration. Fermentation does not have an electron transport chain. Fermentation does not have an electron transport chain. And we're going to talk a little bit about anaerobic respiration, not as in detail as we will fermentation, though. So, anaerobic respiration has an electron transport chain. Fermentation does not. But neither of them use oxygen, and that's kind of where we're at. So, this is what happens when an organism doesn't need oxygen. This is how an organism will break down their food to release energy without oxygen. And if you are interested in shorthand, I almost always in writing shorten electron transport chain to ETC just because it helps shorten things a little bit. I don't have my annotation thing set up. Grab that really quick. There it is. Hit the wrong button. There's that. Okay. That way you don't have to write out electron transport chain every time we're going to talk about it, because we're going to talk about it a lot. So, fermentation is glycolysis and some sort of reaction that regenerates NAD+. So, in order to make NADH, we need NAD+, right? So we need some sort of process that will regenerate that NAD+, in the hopes of making ATP, so all of our energy isn't trapped in that NADH. So our fermentation, any process of fermentation, this is just like the general structure, is glycolysis and another reaction that regenerates NAD+. Specifically, by transferring electrons from NADH to pyruvate, which is our end product of glycolysis, or some sort of derivative of pyruvate. So sometimes there's an extra little step in there. Fermentation is also referred to as an extension of glycolysis that uses substrate level phosphorylation. So when we did cell respiration, we kind of touched on the fact that the electron transport chain uses oxidative phosphorylation. Fermentation uses substrate level phosphorylation. And this is how we generate ATP. We don't need oxygen to carry out, our cells don't need oxygen to carry out fermentation, but they do need sufficient NAD plus in order to generate the NADH. So this is the process of fermentation. It can be broken down into two subtypes, depending on whether or not they are transferring electrons to pyruvate or a derivative of pyruvate. Doing okay so far? All right. So we got glycolysis and a reaction. That reaction's main goal is to regenerate NAD+. They do this through substrate level phosphorylation. So we have two types of fermentation: alcohol fermentation or alcoholic fermentation, and lactic acid fermentation. Those are our two types of fermentation. And we're going to talk about them both. We're going to draw a quick little doodle of the process. So, depending on what we are going to do after glycolysis, that's what's going to help us differentiate between these two processes. All fermentation starts with glycolysis, which is the same process that we talked about with cell respiration. So those diagrams we drew for glycolysis and cell respiration is the first part of this. So the first one we're going to talk about is alcoholic fermentation. And I just want you to write the words down for now. We're going to draw the diagram together on the next slide. So alcoholic fermentation transforms pyruvate, or our end process of glycolysis, into ethanol with a two-step process. So we are going to release a carbon dioxide from pyruvate, and that will get us to a substance called acetaldehyde. Yeah, I might have added an extra syllable in there. And after that, our acetaldehyde is going to be reduced by this NADH. Reduced by this NADH. Remember, reduced means we are adding electrons because we are reducing the charge. And then that will get us ethanol. So the only ATP that we are getting out of this process is from glycolysis. We do not get ATP in the regeneration of NADH or NAD+. So the energy that we would have used in cell respiration that is trapped in this high energy electron carrier is not harnessed in this process. The only energy we get is from the process of glycolysis. Welcome. We're talking about fermentation. Thank you. So this is a two-step process. The first step, after glycolysis, of course, is we are going to release a carbon dioxide from our pyruvate molecule. That will get us acetaldehyde. And then we are going to reduce the NADH byproduct of glycolysis into NAD plus and yield ethanol as a product. So we're going to draw out this process. And I click back and forth for a second. That's all right. Doing okay? Okay. So the first step here is glycolysis. So what do we start with? Glucose. Our net process of glycolysis, yes. Not much. With glucose, or with glycolysis, we are going to take some ADP and some inorganic phosphates. We are going to yield ATP, and that's our energy. And we get pyruvate. That's glycolysis. We know and love glycolysis. We have seen glycolysis a bunch. We are then going to turn pyruvate with the loss of some carbon dioxide into acetaldehyde. But from glycolysis, we have also reduced NAD plus to NADH. But now we have NADH, but in order for glycolysis to keep happening, we need enough NAD plus to keep this going, right? If we don't have NAD plus, we cannot do glycolysis. And glycolysis is where these organisms are getting their energy. So we need to regenerate that NAD plus, which is the whole process of fermentation. And with alcohol fermentation, what we're going to do is we are going to take that acetaldehyde, went the wrong way, and we are going to regenerate that NAD plus and create ethanol. So we've got a little circle here. Keep going back and forth. So we're going to take our NADH, we're going to oxidize it back into NAD plus, and we are going to yield ethanol. As you will find out tomorrow, yeast is a very good fermenter, and it does this process. That is the so has anybody ever made bread from scratch? Maybe. Has anybody seen the process of making bread from scratch? Watched an episode of Great British Bake Off, right? That process, they use yeast to make their breads rise, right? That's because that rising is the carbon dioxide from fermentation creating air bubbles in the dough. So if you open up a slice of bread and see all these little bubbly pockets, it's from the carbon dioxide released in the fermentation process. This is also why yeast is used in the creation of a lot of alcoholic beverages, because that is the alcohol. That is alcohol. Questions about alcoholic fermentation? Awesome. Well, that is one type of fermentation. Now we have the process of lactic acid fermentation, which is only a little bit cut off. There we go. Lactic acid fermentation, we are reducing pyruvate directly. And when we reduce pyruvate directly with that NADH, remember we're all about that NAD plus regeneration, we are going to generate something called lactate. And lactate is an ionized form of lactic acid. Of course it did that. There we go. Lactate is an ionized form of lactic acid. This process does not release carbon dioxide. This process does not release carbon dioxide. There are the same amount of carbons in pyruvate as there are in lactate. You know how some cheeses and a lot of yogurts are kind of tangy? That tang is acid. That acid is lactic acid. Because our tongues perceive acid as sour. So there's not a lot of lactic acid in cheeses and yogurts, but there is lactic acid. And that lactic acid is a product of the fermentation process, because fermentation is used to make cheeses and yogurt. As we know, because we looked at yogurt under a microscope at the beginning of the year and saw all those little bacteria in there, those little bacteria carry out lactic acid fermentation. What's cool is that we can do this too. So, if you were to be sprinting for some reason, whether it be a sport or you're just trying to get to the front of the lunch line, if you're running a lot, so much so that your muscles start to hurt, right? Like you feel like if you cannot stop, if you do not stop, your muscles feel like they are going to quit for you, right? That pain, that burning sensation in your muscles. That is because there is not enough oxygen getting to your muscle cells to regenerate ATP fast enough to carry out the activity that you are doing. So your muscle cells resort to lactic acid fermentation. But it can only do so much of this because that lactic acid burns. And that burn makes us stop, helps us catch our breath, gets our cells oxygenated, and we can pick cellular respiration back up. It is an emergency situation for our muscle cells to help us keep going, to help us make ATP when there's not enough oxygen getting to our cells, which is kind of cool. That we have this kind of fail-safe system. We don't have enough ATP and we need our muscles to get us where we need to go. Questions here? All right, let's do a quick doodle. So, first step in fermentation is, I heard it. It's where we start, glucose. And it undergoes glycolysis. So with our glucose, we are going to break it down into pyruvate. And we're going to yield ATP. We've got our inorganic phosphates. ATP, that's our energy. In this process, we also yield those electron carriers. We go from NAD plus to NADH. But we don't have an electron transport chain in fermentation to get that NADH back up to NAD plus and harness that energy. There's no way to harness that energy, so we just have to essentially get rid of it in another process. In that process, we're going to take our pyruvate. We are going to reduce it. We're going to regenerate NAD plus, and we are going to get lactate or lactic acid. We're all about regenerating that NAD plus in order to keep glycolysis going. Because if we want to do enough glycolysis to make up for the energy we are losing by not having electron transport chain, we got to do this a lot, right? Whenever we talked about cell respiration, we got 2 ATP from glycolysis, 2 ATP from the citric acid cycle, and 26 ATP from the electron transport chain. We're not getting that 26 here. We are only getting this 2. So we have to do this a bunch of times in order to get enough energy to make up for not having that electron transport chain. Which is why this step is important. Because we need that NAD plus in order to keep glycolysis going. Questions here? All right, so we're going to compare them. The title is smaller than the text, but that's all right. We're going to compare these processes. Fermentation, aerobic respiration, and anaerobic respiration. Remember, aerobic respiration has an electron transport chain. Anaerobic respiration also does, but has a different final electronic carrier, and we'll talk about the differences in a second. We're going to start with what they have the same. What's similar with all 3 of these processes? All 3 of them start with glycolysis. All 3 of them start with glycolysis. All 3 of them use NAD plus as an oxidizing agent, meaning it will accept electrons through the process of glycolysis and further down the line. The key difference with all of these processes is how we are regenerating that NAD plus. That's our difference. So that one process we talked about at the beginning of cell respiration is for all of this. Breaking down glucose into pyruvate, generating ATP and NADH. All right. Doing okay? All right. So, we got all 3 of these processes having very similar aspects, and we're going to talk about the differences. In fermentation, our final electron acceptors are pyruvate in lactic acid fermentation and acetaldehyde in alcoholic fermentation. In cell respiration, this is aerobic or anaerobic. We have an electron transport chain. The difference between the 2 of those processes is what our final electron acceptor is. Aerobic uses oxygen, anaerobic uses something else, which depends on the organism. It's not like one thing. So this is where our processes differ. It's in how we get that NAD plus back. Bless you. But there's one other difference that we kind of touched on that I want to talk about. And that is ATP generation. Cellular respiration releases a much higher amount of ATP than fermentation. Because respiration has that electron transport chain that will harness the energy in those high energy electron carriers. Complicated organisms like you and I, multicellular, super complex organisms, need way more ATP than a single celled organism. Cellular respiration allows those complicated organisms to exist because it creates a lot more energy. Usually, organisms that rely on fermentation are much smaller and much less complex than organisms that have cell respiration, because they don't need all of the energy all at once. So, we are going to talk about some organisms. This is some microbiology terminology. It is a way we use to classify organisms based on how they respire, what their metabolic processes are. And these are just two of them. There are a lot more of these labels, but these are the only two that we're going to talk about today. The first is an obligate anaerobe. Obligate anaerobes only use fermentation or anaerobic respiration. They are obligated to do so because they can't survive in the presence of oxygen. There are some organisms that cannot survive in the presence of oxygen. They are obligate anaerobes. Just like we can't survive in a room with only carbon dioxide in it, right? But we also have facultative anaerobes, which are super fun because they use fermentation or cell respiration or anaerobic respiration when they need to. If they can do aerobic respiration, if they do have oxygen available, they will go through the process we talked about last week all day long because it's more efficient. But if they don't have what they need, they will resort to anaerobic respiration or fermentation. But if they do resort to fermentation, if they do resort to anaerobic respiration, they have to consume, or just fermentation, sorry, if they do have to resort to fermentation, they have to consume sugar at a much faster rate. Yeasts and certain bacterias will do this. Yeasts use fermentation. There are bacteria that use anaerobic respiration. Maximizing their chances of survival by having these fail-safes. But if they don't have oxygen, they do need to have enough sugar to kind of make sure that they are getting the energy they need. These are facultative anaerobes, using anaerobic respiration and fermentation when they need to, but only when they need to. This is a really common way that microbiologists classify bacteria. And there are a lot of really cool tests to determine what type of bacteria you are working with. We might talk about some of those later on in this unit. These are two types of respirers, I guess. questions about facultative or obligate anaerobes? I've got one more slide for you, and you have already kind of touched upon it in an exit ticket, right? I asked, what does the kind of, what's the word I'm looking for? No, word's gone. Kind of omnipresent or super widespread use of glycolysis means evolutionarily. So this is, remember, College Board has those 5 big ideas that overarch the entire unit, one of those being evolution. This is our evolution connection. This is how we get from metabolism and cellular energetics to evolution. So ancient prokaryotes, so like our prokaryotes, were thought to carry out glycolysis before the atmosphere was oxygenated. So these bacteria, these prokaryotes that were around, not bacteria, just bacteria, that were around before oxygen was widely available, are thought to have used glycolysis or a very, very similar pathway to get the energy they need. We think this because glycolysis is the most widespread metabolic pathway on Earth. Most organisms do glycolysis. So we think this means that it evolved early, that this process showed up in an ancestor toward the beginning, and that process was efficient enough that it just kind of persisted. Then eventually, photosynthesis would come about, and we'll talk about that a little bit more when we talk about photosynthesis. And that process would oxygenate the atmosphere, make oxygen more widely available, and then things like aerobic respiration could evolve over time. This is our big idea connection. Questions here? Alrighty. So we are going to do a lab over fermentation tomorrow. So I'm going to give you your lab sheet today. It is only 1 page. So you can read through the background and the procedure, as well as answer the pre-lab questions. So I would answer the pre-lab questions on a Google Doc. So you can also do the rest of the lab on that Google Doc. We will be turning it in virtually. But I would keep the lab sheet around so you can take the lab sheet back to your lab stations. That way your Chromebook does not risk getting all fermented, which would not be good for us. So you're going to spend the last 20 minutes. We have a little bit of extra time. That's all right. Reading through this fermentation lab, answering the pre-lab questions, and go ahead and read through the procedure as well. That way we know exactly what we are doing tomorrow. Does anybody have any questions? Alrighty.